All right, so let's go over DNA today. But before we move on to the actual nucleic acids, DNA and RNA, let's talk a little bit about viruses. Since it's this COVID fashionable these days. So what are viruses? Viruses are actually nucleic acids encapsulated within a type of protein or maybe polysaccharide. Now, viruses are non-living things. Why are they non-living? Because they cannot survive on their own. Now, how the hell do viruses survive? They don't really, since they're not really living things, we can talk about survival. We just talk about how they can survive themselves from one person to another person, because they, what do viruses do? They actually infect or invade cells. What cells? It could be any animal cell, including humans. Now, inside these viruses look something like this. Right here, this area or this region is called the capsid. Now, this capsid or capsule right here contains inside the genetic material, which could be any of the nucleic acids, DNA or RNA. Now, here they have this little rod region, and they have these anchors, which are called tails. So what they do, they just anchor this part, they anchor these tails onto the cell, and this part right here penetrates the actual, imagine this would be a cell right here. So it penetrates it, and then the DNA or the RNA, it's released inside the cell when the virus is attached to the host. So check out here how it works. Here you have a cell, boom, it comes and latches onto it, lowers, then it releases the DNA or the RNA, then it's using the machineries inside the cell, the ribosomes, to produce more parts that make up new viruses. Or I mean, a new virus, but the same virus. But what they do, they keep making all these millions of viruses. As they make them, basically, that virus will break down the cells, or the cell will blow up or will break, and then new viruses are released to the surrounding cell. And that's what basically viruses take over the body. This is the short version of how viruses infect the body. Now here you can see HIV virus. This is a copy of how HIV virus looks under the microscope. This would be the capsid right here, and this would be the genetic material. Now, before today, we talked about DNA and RNA in the past. And we mentioned what they're made up of, what they're, besides what they're made up of, we mentioned how DNA, or the definition of DNA, or how DNA looks like. Now, first, DNA, or the first person that talked about DNA was this guy, which was Gregory Mendel. Now, Mendel, what did he do? He was a monk in Austria at a monastery, and he noticed that peas actually, pods, after they plant them, they can produce different type of flower. So he collected the pea pods from a white flower or from a purple flower, actually, and he ended up with new plant that was producing actually a white flower. So he went from purple flower to white flower. So at that time, he didn't know anything about DNA. This was in 1800s. So he called at that time factors, and he said that this function that it's inside the pea pod can control how the future generations look like. Now, DNA, as we know it today, rather than factors, was actually discovered, or was the first person that came up with it, it's about 1950s. So this is more than 100 years later. Now, there's a few scientists that helped, or they participated in determining the structure of the DNA. Chargaff, Franklin, Rosalind, and Watson and Crick are credited as the guys or the people that came up with the DNA structure. Now, it is believed actually that Rosalind Franklin was the first one that came, but these two guys were working at the same university, and what they did, they walked into her office at nighttime, they break into her office and they stole her manuscript, and then they published it ahead of her. And there is actually even a movie about that. So I don't remember the name of the movie, but we can look it up later and I can tell you which one it is. Now, Shargraf was the first one. Now, Shargraf, what he determined is that DNA was made up of nucleotides. Now, there is 4 types. Now, from here on, when I refer to DNA or RNA, I refer as nucleic acids. That's their name, nucleic acids. DNA, RNA. Both of them are nucleic acids. Now, both DNA and RNA are made up of nucleotides. Nucleotides. Now, DNA has, as mentioned before during at the beginning of the semester, ACTG. RNA has ACUG. So if you notice here, T in DNA, it's substituted with U for RNA. Why? Because that's the only way we can know the difference between DNA and RNA. Now, if we look at the structure of the nucleotides, you notice that 2 of them have 2 rings-like structure, and the other 3 have just 1 ring. Now, adenine and guanine are called purines. Cytosine, thymine, or uracil, CTU, are called pyrimidines. You just have to memorize this one or remember this one. So what I remember is I put PAG, purines, oh sorry, PUAG, not PAG, PUAG, that's why I called it. Or a goop, that's why I remember now. Adenine, guanine, purines, PU, purines, backwards. Okay, these ones you can make up your own word, so you can memorize stuff. Also, Shargraf determined that A and T always stick together and C and G always stick together. Why is that? Because he got 2 strands of DNA, or he got a bunch of DNA, he opened it up, and he noticed always when he counted the nucleotides, always A and T were equal, and the number of G and C were also equal. See right here, balance? The number of A and G is the same, the number of GC is the same. The only conclusion that you can draw in this case is that A binds to T and G binds to C. Now, previously, we also, I don't know if you guys remember, but previously we also talked about the type of bonds that exist between these nucleotides. And we call them at that time hydrogen bonds. Now, these hydrogen bonds, there is 2 of them for, and I forgot I messed up here, it should be dotted line. That's how we represent hydrogen bonds. 2 for A and T, and 3 bonds for G and C, 3 bonds. Now let's move forward to Miss Franklin. So this lady, what she did, she was expert in X-ray diffraction. She basically got a couple of fibers of DNA, run them under X-ray crystallographer, and what he did, she noticed that this picture of the DNA showed up like this. Now, if you look here, it kind of represents, or it kind of looks like it's 2 strands right here. So now basically you have all the information about DNA. There is 2 strands made up of a bunch of these nucleotides, and always C and T stick together. And when you look at also this picture, you're going to notice that this is a helix. That's what under crystallography, that's how you interpret this picture right here. There's double strand in the helix shape, with a helical shape. Now these 2 guys, Watson and Crick, in 1953, came up with the structure of DNA. This is their office or the lab where they originally determined the structure of DNA. They had the model up. Now, how is DNA defined as? It's a polynucleotide chain. It's two of them, so it's a double stranded. So it's two polynucleotide chains joined by hydrogen bonding between the bases themselves. These are the nucleotides, basically. It's not really nucleotides, but it's actually the nitrogenous bases that they talk here. So these are the nitrogenous bases that they talk about here, okay? So it's not really nucleotides. This is the nitrogenous bases. They are held together, nitrogenous bases, because as you remember, a nucleotide, it's made up of three things. It's made up of phosphate, it's made up of sugar, and it's made up of nitrogenous base. So here is the DNA, double-stranded, backbone, sugar, and phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And what it holds this whole thing together, it's actually the nitrogenous base pairs, which are what? A, T, C, or G, C. That's what holds the DNA together. So right here you see a polynucleotide that makes up the DNA, helix shape, and there is two strands. Now for RNA, or the difference between RNA and DNA is that RNA has just one. RNA has just one strand. So DNA, it's double-stranded. RNA, it's single strand. Now, since it's double, it's going to be a double helix. All right. Now, when you make a new cell, or you need new cells, because we constantly shed cells, you need to replace them. Now, when we did mitosis and meiosis, we talked about the interphase, or we talked about mitosis, the whole process. So that process was basically when the cell was replicated or a new cell was produced. Now, in interphase, there were three steps. There was G1, kind of like a quick review, S, and G2. Now, the cell was growing in G1, finished growing on G2. The whole cell basically was doubled up. Now, in the S phase, there was one process that was taking place. And that process, it's called DNA, or was called DNA replication. Now, I don't know if you remember, but basically the DNA needed to be replicated at the beginning because it needs to be introduced or added to the second cell. And DNA holds the whole information, or the future information of what cell looks like, what cell produces, and all the functions are controlled by the DNA inside the cell. The DNA, it's basically the CEO of the cell. Now, as the name itself, replication means it basically, you start with one strand and you end up with two strands by replication, making copy. Replication also means copy. For DNA replication, basically, you need a template. So the original strands right here, it's called the template. The final strands, one of them, it's basically, or both of them are basically new strands of DNA. Now, when scientists looked at the replication in vivo or directly inside the cell, they notice that the template, actually, it's not going to be found, or the strands that make up the template are not found together in the daughter DNA or the daughter strands. And just notice that one of them ends up in one strand and the second one ends up on the second strand right here. So if you look right here, this is the original strand. Through replication, this is what it happened right here. You have one new strand, this red one, and the blue original strand right there. So replication, it's actually semi-conservative. Semi-conservative. This is a conservative model, basically, in which this original strand or the original DNA it's found original in the future, in the daughter DNA or the daughter strands. But it does not happen like this, or it does not take place like this. So remember, replication, it's semi-conservative. Semi means half. So basically half old, half new right there. Half old, half new. You can see also right here how the replication takes place. This is just a diagram. First, this is the parental DNA. Then you're going to get something that's going to come and it's going to open up this DNA strand. Now, this enzyme that comes and opens up the DNA, it's called helicase. Once DNA strands are open, as you see them right here, new nucleotides come and they're going to be added. These nucleotides have to be complementary. So if I have A, I bring a T. If I have C, I bring a G. If I have G, I bring a C, and so forth and so on. Now, the enzyme that adds all these nucleotides, it's called DNA polymerase. Now, please just remember this one. That's the name for, there's another polymerase in the cell, which is the RNA polymerase. But remember, for replication, my final product is DNA, and that's why we call this enzyme DNA polymerase, DNA polymerase. Now, at the end, the strands are already formed. I start with one, I end up with two. One is old, one is new, one is new, one is old. That's for the daughter strands. Here you have a diagram of what happened, or a figure of what happened during replication. You get this enzyme helicase, as I mentioned earlier, that comes here and unwinds the DNA. Now this helicase spins so fast because it has to work super fast. And this DNA strand is pretty long. So it spins super fast, actually faster than a jet engine. Unwinds the DNA, then immediately DNA polymerase comes and start adding the nucleotides. Now, one of the strands is copied continuous, it's called leading strand, and then one of them it's copied in pieces because it's moving backwards. And if you're really interested, what do I mean by backwards? I'll explain to you during the lab. But for the purpose of this lab, I just want you to understand this one is called lagging strand because it's copied in pieces, as you see right here. These are pieces. Now, these pieces have to be connected. And there is a third enzyme which is important and which is called DNA ligase. Ligase or ligation means adding or gluing or sealing them. That's what it means, sealing them, okay? One strand, it's continuous, and that's called leading strand. And this is called, in pieces. And this is the lagging strand. Now, if you guys want to watch a video, I got a video right there, and you can do it in your own piece at home when you want to watch it. So one more time, helicase was the enzyme involved in unwinding the DNA. Polymerase, it's involved in nucleotides, new nucleotides attached complementary to the old strand of DNA, right here, and DNA ligase, which seals these pieces of DNA that you're gonna get them when the polymerase moves backwards. Now, before we move into the next step, let's go over the RNA, and then I'll show you, or I will go over the whole dogma of biology. So RNA, or ribonucleic acids, it's a nucleic acid, same thing as DNA, made up of 4 nucleotides, A, U, C, G. So instead of T, as you have for DNA, you have U right here. They have a ribose, which is the sugar, and then the phosphate. This is the nucleotide that makes up the ribonucleic acid. Now, there is 3 types of RNA: M, T, and R. M stands for messenger. T stands for transfer. and R stands for ribosomal. Now, we heard in the past about ribosome. So basically this rRNA is the RNA that's involved in production of the ribosome. Messenger RNA, basically, or the M, it works as a messenger. What do messengers do? They take the message. If I need to send a message, let's say that I'm the CEO of a company and I need to send a message to another worker in the factory, I'm not going to go myself and get exposed or get in touch with a coworker. So what do? I have a secretary, I have somebody to send them to do to give the message out. Now that is called a messenger. That's exactly what it happens inside the cell too. So that's why this messenger is there. And the last one, it's transfer. Basically, that transfer RNA, we're gonna learn in a little bit. What they do, they just basically bring the amino acids to form the protein. Now, since I talked about everything, all the components, let's talk about dogma of biology. Dogma of life, biology, biochemistry starts with DNA, then you have the mRNA, and then you have protein. So the final product, the final product that you need or everybody is made up of, it's called the protein. DNA, it's basically the code, the blueprint. Now, to get the message that's on the DNA to be turned into protein, we have this intermediate, which is this messenger right there, the messenger RNA, because the message has to be taken out. These two are stuck inside the nucleus because that's where DNA is found. So this process right here, which is called transcription, takes place inside the nucleus. The process of producing protein, which is called translation, takes place on the ribosome. Remember from the past when we talked about the cell, ribosome synthesize protein. So ribosomes are important because they make the final product. So this is the whole dogma of biology. Also biochemistry. Also DNA turns into DNA when a new cell is needed. This is basically for new cells. If you lose a cell underneath comes up and it's replaced with a new, the cell that you lost is going to be replaced with a new cell. This process, as we learned about a couple of minutes ago, was called replication. Now, replication takes place during mitosis. Remember, replication takes place during mitosis and meiosis also too, but mainly to make a new cell, the process takes place during mitosis. All right, messenger RNA, as I mentioned earlier, produced in the nucleus. The process is called transcription. Now, the messenger RNA carries the information from DNA to cytoplasm where the ribosomes are. Now, as you get to the ribosomes, you get the transfer RNA, and this transfer RNA carries the amino acids. In the past, we learned that proteins are made up of amino acids. So to make a protein, I need a bunch of those amino acids. Now, there is 20 amino acids, 20 total amino acids. That's what we learned. 20 amino acids. So basically, I have to produce different types of or different tRNAs to carry these amino acids to the site of translation or on the ribosome. So basically, transfer RNA carries the amino acids. They deliver them to the site of protein synthesis, which is the ribosome. Now, there is 20 amino acids. So theoretically, I would need 20 different types of tRNA 20 different types of tRNA to carry those 20 amino acids. Now, there is one problem. Oh, let me go quickly over ribosomal. So ribosomal, as I mentioned, also produces the ribosome. Now, there is a problem when we talk about transcription. What the problem is? The problem is we have 4 nucleotides. Now, when the code is read, or these nucleotides are read, or the nucleotides make up a very long sequence. Let me show you an example. This would be a sequence that you would have. This is an mRNA sequence. Now, how the heck this message is read? The message is read 3 letters at a time. 3 letters at a time. Now, since it's read 3 letters at a time, and there is 4 total nucleotides, there is a problem. I have more than 20 codons, because this 3-letter, it's called a codon. So I have more than 20 codons that code for amino acids. Remember, there is only 20 amino acids. Only 20 amino acids. But when I'm going to arrange these nucleotides, which are read 3 at a time, and that's called the codon, I'll end up with actually 4 to the power 3. This is basic math. 64 different arrangements for these nucleotides, these 4 nucleotides. So now that sounds like a problem. All these codons right here will code for an amino acid. Now there's a little exception. There's a little exception. There is 3 codons right here. And you don't need to remember them. You just need to remember that there is 3, which code for stop codon. By stop, it means that there is no amino acid to be brought in there. So remember, this is the codon, and then you bring the amino acid based on this codon right there. Now, these 3 codons are basically do not code for amino acids. So then there is actually a total of 61 codons that code for amino acid. Okay, now let's go. Oops, stop share. Let's share it again. Now, let's see what's happening here. This thing doesn't want to work now. Okay, let me go back again here. Oops, what's going on here? No annotation. Clear. Drawings. Okay. Let me see what the heck is going on right here. Oh, don't tell me this thing froze, please. Holy schmoly, that's all what I didn't want. All right, I guess I gotta come back. This thing froze, damn it. Freaking technology. Hey, I gotta come back. I think this thing froze, guys. So let's go back here. So there is 64 codons, 61 code for amino acids, and 3 of them are stop codons. So if you notice here, there is a problem. There is 61 coding for amino acids, but there's only 20 amino acids. So how can I please all these 61 codons to code for at least one of the amino acids? So if you look right here, you're going to notice that there is more than one codon. Like an example right here for leucine, for example, there is 1, 2, 3, 4, 5, 6 codons for leucine, which is one of the amino acids. There is 6 codons coding for it. There is six codons coding for it. If you look at histidine, there's actually 2 of them for histidine. 2 codons. So that's how we solve the problem of getting all these 61 to code for amino acids. Now, these large numbers of codons coding for one amino acid, it's called redundancy. Redundancy of the code. It's called the redundancy of the code because you use different codons to code for the same amino acid. So if you notice right here, this whole table right here, this is called the genetic code. Now, you would never have to remember this one because I don't know this one by heart. I just always go to Google or I go everywhere else. If I give you a question in the exam, I will give you the code. You just have to know how to read it. So these 3 nucleotides right there or letters make up the codon. That's how basically, or those are the words that are used for the genetic genetic makeup or genetic coding. Now let's go back right here. As I mentioned earlier, when you make protein, you have to convert a nucleotide to an amino acid sequence. And then the codons are the words that make up the whole sentence, the language itself. Now the process through which the message it's produced, or it's turned from RNA to protein, it's called translation. And the message which go from DNA to RNA, it's called transcription. Now let's go quickly over transcription. Transcription, basically, it's when you go from DNA to RNA. As you notice right here, DNA, it's double-stranded. RNA, it's single-stranded. Now, if you notice also another feature of RNA that you're not going to find on the DNA, there is the U versus the T. Now, this one right here, it's the strand that's going to be transcribed. So if you notice right here, this strand right here, it's basically the complementary of the strand that's going to be transcribed. Once you get the RNA, you're going to end up actually producing at the end doing translation, the process of translation, you are going to produce, or you're going to make up the protein. Now, there's one more feature that I forgot when I talked about the genetic code. There's always a start codon, and the start codon starts with AUG, and it's called methionine. So let's see what happens during transcription. There is 3 stages for transcription: initiation, elongation, termination. Initiation, you have to do something to initiate the actual transcription. Elongation, it's when you go from one nucleotide to many. This is elongation. And termination, it's basically you're going to encounter somewhere something at the end at which the strand it's already produced. Now let's go over each one. If you look right here, step 1 involves the enzyme RNA polymerase. Remember, we talked about DNA when we talked about replication, but RNA polymerase, what it does, RNA polymerase, it makes RNA. That's why it's called RNA polymerase. Now, this RNA polymerase, during initiation step, it binds to this region right here, which is called promoter. Binds to the promoter. Now, once it binds to the promoter, it starts moving up. While it moves up on the DNA or upstream, it unwinds the DNA inside. It unwinds the DNA temporarily. Why does it unwind? Because while it unwinds, it keeps bringing these nucleotides and keeps matching them with the original strand or the DNA or the DNA that's going to be transcribed. This step right here, it's called, while it's moving along, it's called elongation. So the first step, it's initiation, where it binds to the promoter region and it just barely start moving. Elongation, it's when it starts making the RNA, basically producing or adding the nucleotides and binding them. And the last step, it's called termination, where it encounters a terminator region right here, the terminator region, and basically it knows that the RNA or messenger RNA, it's already formed. Now this process, if you guys could remember, since DNA is stuck or it's found in the nucleus, this whole process of transcription takes place in the nucleus, not nucleus, in the nucleus, okay? Now, let's see what happens with this messenger RNA. When we talk about DNA, as it's seen right here, DNA, it's made up actually of a few regions, two regions. We call them intron and we call them exon. Intron, it's also known as junk DNA. Exon, it's basically coding. So exon, it's basically this region right here that make up the actual nucleotides, which will make up which is used to make up the protein. Now, when DNA is copied, you just want to copy only the exons for any of the genes. The problem is what? You have these introns that are intercalated. Now, when RNA is copied, RNA polymerase cannot just jump from one exon to the next exon. It does not jump, but rather it copies the whole message, including the junk DNA, including the parts that are right here. Now, if you notice right here, this would be the raw mRNA, it's called. This is the raw mRNA or raw RNA. Now what happens, one of these enzymes, which is called spliceosome, and you have it, oops, right here, I thought that I put it somewhere, but I guess I didn't. This enzyme, which is called spliceosome, which comes and recognizes these introns regions, and it cuts them like scissors. And now you end up with what? With the actual mature RNA or mRNA. Now, this is when the mRNA is ready to leave because it has the coding sequence. It does not have anymore the junk DNA. Now, this mRNA, it's going to leave the nucleus through this region, which are called pores, and it's going to head for what? For the ribosomes. Here, you could see the whole process. This is basically transcription. Remember, it takes place in the nucleus, transcription. Once you get the mature RNA, this is the mature RNA, boom, it squeezes out and it's going and looks for ribosome, where the next process, which is translation, will take place. You have a video right here, a link that you guys can use to actually look at actual transcription. Now let's go on to the next step, which is actually translation. Let's see what happens here. Now, translation follows immediately transcription. Now, for translation to take place, you require the transfer RNA, which we talked about earlier. Now, a transfer RNA has a couple features. Has this top right here, where basically the amino acid is gonna bind hydrogen bonding to the tRNA, and right at the bottom right here, it has a region which is called anti-codon. Now, anti means it's against. Now, this anti-codon, it's complementary to the codon. So if I have a sequence right here, like the one that I had earlier, AUG, this is what is going to basically be complementary. So this would be what? UAC. So the anti-codon, we're going to be UAC, the codon would be AUG. And then you have the actual codons that follow from the mRNA. But basically a tRNA, two features that I want you to remember. A region where the amino acid is going to bind to it, and then the region which will bind by hydrogen bonding to the mRNA. So basically what's going to happen, this is the same thing, the tRNA. Basically what's going to happen, you see how these tRNAs temporarily bind to the mRNA? So basically this tRNA has this anti-codon region, which is complementary to the codon. So remember, mRNA holds the codon, tRNA has the anti-codon. Now, earlier we left off when the actual mRNA was squeezing out of the nucleus and it was heading for the ribosome. Now, this is how a ribosome looks like. Now, a ribosome, it's made up of a large subunit and a small subunit. These two are, as long as they are not functional, they are separated. Why? Because you don't want to use a lot of energy, you want to conserve energy. To hold them together, it requires energy. But if there is no need to translate, then they don't stick together. These two come together only when translation is ready to start. Now, if you notice here, this large subunit has these regions right here, these compartments, which are called tRNA binding sites. So basically, tRNA is just going to come and squeeze right in here. Now, here also has a hook. This hook right here on the large subunit, it's used for mRNA to bind, and then mRNA is squeezed forward right here. So you can see right here, mRNA, it binds, the hook is, let me see, based on this picture right here, the hook would be right here. You can't see it really well here, but it's gonna be somewhere here. And now it's moving forward. Or actually, I'm sorry, it's the other way around. It's moving like this. Now, how does, or what happens during translation? This is the mRNA, which contains the genetic code. Now, remember, the start of genetic code, it's always what? A, U, G. Now, all these ones can be any other nucleotide possible there. In our case right here, let's see, what do we have? Well, we don't have any right here, but let me label them. U, A, G, C, G. I'm just gonna keep going. And right here at the end, you have a U, A, G, which is one of the stop one. I think it's one of the stop codons. I don't really remember, but I think it's one of the, or UGA, it's one of the stop codons right there. Now, basically, this mRNA, it's gonna come and it's going to attach to the small subunit, to that hook right there. Now, it's gonna get pulled until in the first compartment right here, or the region where the first compartment matches the small subunit, the first codon shows up, which is AUG. The moment that the first codon it's here, the first tRNA comes, if you notice right here, UAC, which is the anti-codon, binds through hydrogen bonding to the mRNA, and brings the first amino acid, which is methionine. Immediately, as this amino acid settles in there, immediately as the tRNA settles in there, the large subunit comes and it will attach to the small subunit. This process is called initiation. Next step, what is going to happen, it's elongation. Now, there is an empty unit right here. So the next tRNA that matches exactly the codon right there, whose anti-codon matches this region right there. The codon brings the next amino acid, comes, sits in, and then these two amino acids will stick together. You can see it right here. So this is how it forms. The next tRNA comes in, amino acids are set in. Now they're going to form the bond right there. Now what's going to happen? As soon as this amino acid or polypeptide that forms here, it's going to attach, then this tRNA is going to be transferred in here, and the one from here is going to get kicked out. Then here in this region, you're going to have the new polypeptide. Now that's why this compartment right here, it's called P, or the one that makes the protein. The one that's going to be always empty, or not always, like the one that always going to receive the new amino acid or the new tRNA that brings the new amino acid, it's called A, or amino acid. So you have the two compartments. This is the P compartment right here, or the P side, and this is called the A side. You can see them right here. P because that's where the protein grows. A because that's where the tRNA always comes. So remember, this A site always receives the tRNA. Now, if you look at this one, the cycle or this elongation keeps taking place constantly until in the A site right here, you're going to encounter what? The stop codon. The moment that you encounter the stop codon, there is no anticodon or tRNA that codes for this or that matches this mRNA right there, the stop codon. And then what's going to happen? This this tRNA is going to be kicked out, and then there is nothing to fit in. The moment that this tRNA is kicked out, this protein right here, this peptide, it's released, and then this ribosome will disassemble. Now, the process I just described, at the end, it's called termination, or that's the last step when it encounters the stop site. Now, in reality, actually, you're gonna have many of these ribosomes attached on one piece of mRNA, on this one piece of mRNA. So you're gonna have many of this one attached to this piece of mRNA right there. Now, this whole structure right here, it's called the polysome. Why you have so many attached at the same time? Because it's called time or efficiency. If I would have just one ribosome constantly translating, it will take a long time. But if I have many of these ribosomes or this RNA, mRNA attaches constantly to a new ribosome, even it did not leave the other ribosomes, I'm saving time. Most of the time you have between 10 and 20 ribosomes attached to the same tRNA, mRNA. Now here you guys have the actual video, and you can watch or you can look at translation. Quick review. What does translation represent? As I just talked earlier, or I just mentioned earlier, it's this process right here, RNA to protein. Where does the information for producing a protein originate? It's not on the RNA, guys. That's the messenger, basically. That's the part that's going to bring the message. The key part here, originate. It's on the DNA. And the last one, which one has a linear sequence of codons? If you looked at it, mRNA is the one that has the linear sequence of codons. This could be potential question or potential critical thinking question, or how to approach the questions that I ask. Now, during these processes that we talked about it, and we talked during mitosis too, we can encounter mutations. What is a mutation? Mutation, it's basically when you add the wrong nucleotide. If I come and I have this message right here, and instead of an A, I'm going to put a G, I am going to end up with the wrong amino acid and eventually with the wrong protein. I don't know if you remember on lecture 3, I gave you, or I showed you what happens when you have sickle cell anemia. The cells look totally different, the blood cells. Why? Because of just one single mutation. And you can see it right here. There's only one single mutation between the normal hemoglobin and the sickle cell. It's CTT. This T right here is replaced with an A. Now you completely change the actual shape of the cell and you end up with sickle cell. Now this process through which the wrong nucleotide is attached, it's called mutation. Now mutation is very frequent during replication. It happens 1 in 100 million cell division. And remember, we make millions of cell divisions we're gonna have because we constantly replace cells, and we are made up of a bazillion of these cells. Now, if mutations take place, there is a problem. Why? Because you can end up with diseases. Now, remember how did we call the process, or the process through which the cells are sent to commit suicide, or the cells are killed in during mitosis? I don't know if you remember, but it was called apoptosis. And remember also, we had checkpoints to make sure that the cells, or the cell that's going to replace the cell that was lost, it doesn't have any mistake. If it escapes those checkpoints and it's not committing suicide, it's a problem, because that new cell can eventually get the message to divide, and once it divides, many cells after that will have exactly the same message or the wrong message. And that can lead to major problems. Most of the time, it's cancer. And remember also, we talked about mitosis, those cells that have these mutations, they become cancerous. Why? Because they divide so fast. Now, there's different types of mutations. There is base substitution that we can see, deletions, or insertions. All these mutations can be caused by mutagens or by spontaneous mutation during mitosis. What is that? Spontaneous is basically when errors take place during replication, but mutagens are basically radiation, radioactivity, and chemicals. Chemicals can replace some of those nucleotides because they mimic or they look like it, so then they can be replaced. These are technologies the big pharmas take advantage of when to produce medications or to produce treatments for different types of cancers. Now, what the heck is base substitution? Substitution, it means that you replace one with another one. T replaced with A, it's called base. You just substitute one nucleotide, it's one base. Now, the problem is, if you change the amino acid, it's a big issue, because this could lead to different shapes or different structures. And this eventually can lead to the actual cells not functioning properly. Now, this is the least out of all the mutations, the least one, the one that would give you the least problems. Why? Because sometimes due to redundancy of the code, and remember that many codes or many codons, at least 2, 3, 4, 5, 6 code for the same amino acid. Sometimes if you're lucky, this mutation might give you actually the same amino acid. In this case right here, it didn't, but it could happen due to redundancy of the code. Even though you have a mutation, you can end up with the same amino acid, and then it's not going to affect at all the actual cell. The problem is the next two of them, deletions and insertions. Here is a normal gene. If I come here And let's see, U. Right here, I am going to take this nucleotide out. This is a deletion, example of deletion. What's happening right now? If you notice, this whole codon right here will shift to the left. This whole codon right here, now it's going to shift. And I change the amino acids. This codon right here, it shifted. Instead of being U, G, G, C, G, C, A, now I have what? If I replace this one, I have U, G, G, C, G, which is basically coding for a total different amino acid. So if you look right here, you have methionine, lysine, phenylalanine, glycine, alanine. When you go right here, you have methionine, which is fine, lysine, the second one. But the third one, instead of being phenylalanine or PHE, it's leucine. The fourth one, instead of glycine, is going to be alanine. And the last one, alanine, becomes it becomes histidine in this case right here. Now you completely change the whole protein. This is a big issue. Now it would be exactly the same analogy if you would do, or if you would have insertion, with the exception that now, if you put a nucleotide right here, it's going to shift to the right. So now, instead of having, for example, U, G, G, C, like here, right? U, G, 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 C, G, C, A. Now let's assume that you insert one. So now you're gonna U, A, G, G, C, G, C, A. Now when it's actually translated, you end up with different codons. So instead of having instead of having U, which is kept right here, GGC, you have AGG right here, which replaces this codon right here. Now, there is a problem because you completely change what? The actual amino acid, and the protein will come out different. Oops. Now here you guys can see some effects of mutation. A popcorn kernel, or a kernel that's white for the corn, and this is the purple one. This is the normal gene. This is actually a mutated gene. So the white one, it's a mutated one that we buy. Now, how did scientists can perform this mutation? We can perform this mutation actually in lab, not only in vivo, basically during mitosis or during live replication. We can do this one in the lab through this process called DNA technology. Now, DNA technology applies genetic engineering, which involves cloning. Cloning, it's basically when you take a copy of one gene and you insert it into another organism. This is called recombinant DNA technology. So cloning, it's basically when you get copy of gene from one organism and you insert them into another one. I don't know if you guys ever seen, but there is out there blue roses. Now, naturally, they don't grow in nature like that. Through genetic engineering, especially cloning, we took the blue gene from another plant and we actually inserted it into the rose, and the rose now it looks blue. It acquired the gene and it turns blue. Now, for the process of cloning to work properly, to get the final product, which is the recombinant DNA, we need a few structures. We need a vector or a plasmid. We need a restriction enzymes, and we need a DNA ligase. Now, what each one does. A plasmid is just basically just a small circular piece of DNA that it's found inside the bacteria. So right here, this would be the plasmid. Now, why are plasmids so cool? Plasmids are so cool because these plasmids have the ability to divide on their own. They don't need or they don't interfere with the genomic DNA of the bacteria. This is the bacterial chromosome. It's not affected. This is just an individual piece that will not affect or will not affect bacteria at all. Bacteria is kind of stupid. It doesn't know that I use its own plasmid to make a cloning or to make whatever I want to do with it. So how do I do this one? I get this plasmid outside from a bacterium, and then I'm going to isolate a gene. In this case right here, let's assume this is the gene of interest that I'm thinking of. Let's assume that this is the gene for insulin. Now I want to produce insulin in the lab. Back in the days, insulin was extracted from pigs, but these days, actually insulin is produced through the DNA technology process. So that's why it's a little bit more expensive. Now, what you do here, you're going to come and you're going to cut this plasmid and you're going to cut this gene out using the same restriction enzyme. Now, what's a restriction enzyme? As the name tells you, it's basically restricted and it recognizes only one side. Basically, restriction enzyme cut the plasmid in the foreign gene at known sequences. They recognize that sequence. Now I have to use the same restriction enzyme when I splice my gene of interest, and the same thing when I splice the plasmid. So if you notice right here, you see these two ends or these ends look the same. Why is that important? And it mentioned right here with the same enzyme. Why is this important? Because I have to match them perfectly in here. Now, boom, they come, they match perfectly. The next process is where you're gonna use the DNA ligase. It's the same type of ligase that was used when we talked about replication. So we're gonna get this DNA ligase, which will come and seals these ends right there. Now the ends are sealed. Now I take the plasmid, I put it back into the bacteria, then I allow bacteria to multiply, or I can tell bacteria to start expressing or producing the gene itself. In this case right here, what I do, I multiply this, I reproduce this bacteria or multiply it, and then eventually I'm gonna take these plasmids out, cut the genes, and now I started with, at the beginning of the process, I started with one. At the end, I have millions of it. Now, this one can be used basically to shoot them inside plants, and you can mutate them. It's not used too much. This technology is not used actually in humans or animals because it's not 100% efficient. It's not even close to 100% efficiency. So then you might end up with mutants, but it's used in plants. Then also, as I mentioned earlier, I can tell these bacteria to rather express those proteins for me, and I end up with the actual protein. I harvest my protein. Products of DNA technology, as I mentioned earlier, insulin, HGH, and vaccine. Vaccine is another product of DNA technology, and as you guys hear probably on the news, they tried to come up with this vaccine for COVID-19. Actually, this is one of the technologies that they use. I don't know if you guys heard about Moderna. Moderna uses a different type of technology. They use the mRNA technology, which matches the DNA, but most of the companies, they use this type of DNA technology. When you use DNA recombination, we can produce genetically modified organisms or transgenic organisms. Now, what is the difference between them? Genetically modified, basically, I take the organism and I introduce artificially another gene on it. And when it's transgenic, this one, it's kind of like genetically modified, but it's transgenic because I take it from one species and put it into a different species. Now, we produce a lot of genetically modified plants. Why? Because we can make them resistant to herbicides, pests, and improve the nutritional value. And this is one of the, I actually, when I did my research for my master's and PhD, I worked in basically increasing in rice the nutritional profile. GM animals use improved qualities, production of proteins, or therapeutics. That's what we could use them for. And the last step that we can use for, in which we can use recombinant DNA technology or cloning, It's called polymerase chain reaction. Now this one, it's used these days mainly, it's used actually in research mainly, but there's used also in courts. Now, polymerase chain reaction stands for you take one strand of DNA and in the lab using a machine, which is called a thermocycler, you can produce millions and millions going through different cycles. So polymerase chain reaction, method of amplifying or making copies of a DNA molecule, segment only, not the whole DNA, specific genes of interest. Now, for PCR, I need a couple things. First of all, if I need to make a new piece of DNA, I would need an actual DNA polymerase, the same polymerase that was needed during replication. Also, I need one more thing, which are called primers right there. Now, the first thing that you're gonna do when you use the PCR, you're gonna get your strand of DNA. You went to the crime scene, you found one piece of hair with a follicle. Okay, remember, it has to be a follicle. Now, you take that piece of hair, you take it to the lab, and now you just have one strand of DNA. That's all what you have. Now, using this technology, you're able to make millions of these strands at the end, millions of these strands, and then eventually you can match to the suspect. How do we do that? We get that machine, we put that, first we isolate the DNA from that one cell. Then we're gonna go put this strand of DNA in the machine in a tube, and then we heat it up to 95 degrees Celsius. At 95, DNA strands separate, and now I have two single strands right there. Next thing, I add this small region, which are called primers. These primers basically match perfectly the DNA. I make them, I order them from companies that make them. Once these primers are added, and this one takes place at about 55 to 62 degrees Celsius. Once these primers stick to the complementary region, DNA polymerase, which is the next enzyme, comes and elongates it in both directions. At the end of the first cycle, going through heating, cooling to 55, 60, and then heating up to 72 degrees Celsius where the elongation takes place, I end up with two strands of DNA, 1 and 2. Next thing, cycle 2, go back to 95 degrees Celsius, 57 degrees Celsius or whatever, let's put 60 degrees Celsius, and then 72. So at the end, now at the end of the cycle, I have 4 of them. At the end of cycle 3, I have 8 pieces, and so forth and so on. So I start with 1. By the end of the third cycle, I have 8. Usually we run between 30 and 40 cycle. At the end of the 30th cycle, you have about 16, 18 million, something like that. You guys do the math. If you notice, they constantly double. 1, 2, 4, 8, 16, 32, 64, 128, 256, 512, whatever it is from there on. Now this DNA that it's amplified, or this piece of DNA that is amplified, then it's gonna be run on the gel right here, and each strand or each gene has a different size based on the number of nucleotides that it makes. I can have a gene, let's say gene 1, that it's made up of 250 base pairs or nucleotides. I can have gene 2, which is of 400 base pairs, and I can have gene 3, which is made out of 800 base pairs. Now I take those products that I get in the tube, and I'm gonna load them up in the gel right here, and then I apply electrical current, negative at the top, positive at the bottom. Why? Because DNA, it's negative charge. It has a negative charge. Now, once I apply the positive, based on their size, they start separating the way you see them right here. They start separating. The larger ones, the 800 base pair, move slower. The lighter one, or the shortest one, will move much, much faster. Now, at the end, you end up with a complete gel, which it shows the size of each of these genes. Now, this technology could be used in this process that's used by courts for a paternity test and for crime scene, which is called DNA fingerprinting. This is basically each one of us have genes that are different in size. Now, for this paternity test or crime scene test, they use 12 genes. Now, in general, they use only, this is the max number, but in general, they use about 5 genes to save time and to save resources. Because when you run 5 of my genes that were found to be different in everybody, with 5 from, let's say, Jimmy and 5 from Jordan, when you separate them here, we all look different. Some of us might be the same, or they travel at the same level. Some genes might be about the same size, but all 5 at the same time, it's impossible. The only way that it could happen that all 5 are the same if we are related. That's when I'm going to go back to 12 genes, because I want to see a complete spectrum of it, all 12 genes. Now, even if I'm first degree cousin with somebody else, or I'm brother and sister or brothers, we do not have the same the same size for all 12 genes. The only time that this could happen is if we are identical brothers, because identical brothers have the same DNA. Now here you guys have an example. DNA was isolated, DNA was amplified in this tube right there, then this DNA was run on the gel. Suspect 1, suspect 2, DNA collected from the crime scene. This is the actual, how the DNA look when it's separated. 1, 2, 3, 4, 5 genes. Suspect 1 here, when you look at it, this one, they do not match. This one kind of matches this one right there, but it's only 1 out of all 5 that match. So this could not be the suspect that was at the crime scene. Now, when you look at suspect 2, what do you notice? All of them match the crime scene. And now if they match the crime scene, what's going to happen? This is the suspect that actually, whose blood was collected from the crime scene. So this could be the potential criminal or potential perpetrator, whatever it was the case. So basically this technology, it's very, very efficient. First time, I think it was used in the O.J. Simpson case. And at that time, I think that was the first time it was accepted in the courts. But these days, it's actually used for everything. And you guys hear that district attorneys go back and they have stored samples from 60s, 50s. And then a lot of these people, they are in jail for like 30, 40 years, 20 years, 15 years. They were incriminated. Now they just get actually, they are let go, because due to this advancement in technology of using recombinant DNA, it's not anymore, or it doesn't work anymore based on, hey, you know, I saw Jack that was at the crime scene, so I'm assuming that he killed the person. That just because I saw him, or potentially I could just lie. We go based on the facts that are collected from the crime scene. And this is all about DNA.